basic operations of life. Examples include the direct formation of the cytoskeleton that forms the structural foundation for cells and those that control cell division. In general, any change to a DNA sequence that affects the function of the gene it comprises should affect the organism one way or another. Mostly the change will be for the worse. You simply cannot tinker with something as intricate as the instructions for life without messing something up. It will probably be something important. This is why most mutations to functional genes are thought to be harmful. Studies estimate that as many as 75% of mutations that swap one DNA base for another within genes cause some sort of reduction in survival or reproductive output. Though most of our genes predate the origin of complex life, others we picked up and tweaked along the way, like genes that allow us to breathe oxygen or perceive light. We eventually gained versions of genes that control the formation of our bodies, with a head at one end and an anus, thankfully, at the other. Much later, we acquired genes that covered us in hair, until changes to those genes took much of that hair away. More recent acquisitions include gene variations that govern complex patterns of thought and reasoning, as well as versions of genes that allow us to communicate those thoughts with one another through speech and symbols. Indeed, our bodies and our genes are a living testament to the lives of our ancestors, and so the deciphering of the human genome promised to provide new insights into our evolutionary past. We could find out how, exactly, natural selection has operated and which genes it has affected. We could look for clues about what genetic changes make us different from our primate relatives and when exactly we acquired them. We could piece together our history so much more thoroughly than would ever be possible with fossils and artifacts alone. The technology behind the sequencing of the human genome advanced quickly toward the end of the 20th century. As an undergraduate student at the University of Illinois in the mid-1990s, I worked in a research lab where, after first paying my dues by washing lab equipment, I was trained to sequence DNA. The method we used, first developed by British biochemist Fred Sanger in 1977, came to be known as Sanger's chain termination method, which earned him his second Nobel Prize. Sanger's method included copying sections of the DNA molecules and pasting them into bacteria, which conveniently make lots of copies of themselves, and thus also the copied DNA, simply by growing in a petri dish. The large amounts of DNA they produce could then be isolated, purified, and sequenced. Reading the DNA sequence was the trickiest part. I had to first make a very thin gel by positioning two rectangular sheets of glass separated at the top by a thin piece of plastic and then carefully tapping the other three sides to prevent leakage. I would then heat a gel solution in a microwave oven and pour it into the top of the rig, in much the same way you might pour Jell-O into a mold, and then let it solidify as it cooled. Inevitably, a small air bubble would form somewhere in the gel, which I would carefully try to push toward the top. If I failed, that part of the gel could not be used. The DNA molecules that had already undergone the chain termination procedure would then be injected with a syringe-like pipette into narrow channels at the top of the gel, which was positioned upright with the top and bottom sitting in a vat of liquid so that an electric current could pass through the entire apparatus. Because DNA is negatively charged, the fragments would be attracted to a positive charge at the base of the gel, forcing them to slowly pass through the gel. Smaller segments of DNA would travel slightly faster, so by the time I shut off the electrical switch after a few hours, the DNA fragments had arranged themselves neatly according to size. Dunk the gel in a tub containing a radioactive isotope that would stick to the DNA, and the fragments would appear as glowing bands on an X-ray film. Because each column corresponded to one of the four nucleotide bases, adenine, thymine, guanine, or cytosine, my job was then to simply determine the sequence of bases by starting at the bottom of the gel and moving toward the top, recording which column contained a visible band. This was